these photographs and stills and video, sometimes depending on your software, to stitch them together. There are softwares that recognize the overlapping portions of photos, and essentially what you can do at that point is stitch them together. It's called structure from motion, and this is the really simplistic way of talking about it. You're essentially stitching it together, so it creates within this software program a 3D model that you can twirl around, zoom in and out of, and it's a really great way to be able to capture images of a sponge on a rock, or if you're looking at a bigger boulder with multiple organisms on it, or if you're going to be doing a habitat mosaic, it's a great way to get almost like volumetric 3D measurements of what you're looking at that we're hoping to use for long-term monitoring. So if we were to go back to this boulder or to this area of habitat, are there any changes? Did they grow? Did they get damaged? Did they change in size and number? Are there new corals and sponges? Are there fewer? And it's a really cool way for education and outreach products to make those products, in addition to cool ways to get at some of our research objectives, which is long-term monitoring. And if anyone wants an example, the Nautilus collected imagery in the way that I'm describing for the Davidson Seamount whale fall in the Monterey Bay National Marine Sanctuary. And the folks from the sanctuary worked with the Monterey Bay Aquarium Research Institute to create this photo mosaic. So if you go to Sketchfab, sketchfab.com, it's a public website where the researchers from MBARI and Monterey uploaded the whale fall 3D mosaic. So if you just type in the search bar on Sketchfab, Davidson whale fall, it'll pop right up and you'll see it's on MBARI's profile. And they have a bunch of different other photo mosaics they've done that you can go look through and zoom in and zoom out and twirl it around to your heart's content.
Thanks so much, Lizzie. And I know you've done some photo mosaics in Channel Islands, so those will also be available in the future in the next 6 or 12 months. I'm not sure how long it takes to get them processed, but there's enthusiasm for seeing those.
Yes.
So we've been seeing, while we've been doing this, our cruising along this wall here, we've been seeing many of those king crabs, and they're accumulating in small areas. And so Mary Wickstein was commenting that it generally means that they're getting ready to court and mate. So we've got that going on.
There's a question about, has Nautilus been here before? And are you comparing it to prior visits?
So the Nautilus has been to Footprint Reef before. They, I believe, approached from the northwest side, and we're approaching, or excuse me, the northeast side, and we're approaching from the southwest side. So our goals might be a little bit different, but I think at some point we could revisit some of the areas we were. In NA123, I believe, was the mission that they were here. But we also have a bunch of other footage and transects done in this area from other surveys in addition to the Nautilus that we could compare this video coverage to. So I think, yes, that's one of the objectives here, is because this is such a well-surveyed area. Are we noticing any changes going on? And in addition, we're much deeper on this side than other surveys have been in the past. So we're extending our range of knowledge in terms of what we're finding and expecting to find here on the deeper slopes of this feature.
I'm gonna grab Bubble for a second. Yep.
Great questions, everyone. Keep them coming.
Okay, there's, I can keep doing that. There's
Yeah.
someone who notices a lot of yellow, white, and various shades of red, and wanting to know what is the significance of those colors. Is there an evolutionary advantage to those different colors in the deep sea?
Well, I do know that those wavelengths are the first to be absorbed. And so it could, help them be more cryptic into the habitat that they're occupying at these deeper depths. I mean, this is no light areas here. I mean, you're also looking at, then you have other adaptations with bioluminescence and that can occur at these deeper depths. But just thinking about how light refracts when it works through the water column, and that's why the oceans are blue, is because that's one of the longer wavelengths that's not absorbed as quickly.
Yeah, so it could be that, like, for example, rockfish are red because they're hiding from predators, or they're using it as a way as camouflage because they can't see red at this depth. They kind of blend in with the brown rocks in the background, but that's not something I'm super well versed on. But I believe, but thinking back to my general courses back in college, those were some ideas that we were talking about in terms of coloration, especially in the deeper areas of the ocean.
Still moving?
There's some more of those clams we saw earlier. Do you remember what they were called? I'm looking them up again. Yeah, let me just. They're beautiful.
Or
Yeah.
I believe is the genus.
Yeah, Acesta clams, and we have those sorts of. They're beautiful. Yeah, just to take a look because they're different looking. They belong to the Yymmedd family. They're called flame scallops. Notice the very long tentacles, very diagnostic. And there are similar species found in the Gulf of Mexico. That was from an earlier conversation from Mary Wixton.
Can we zoom in now?
Are they always found in pairs like that?
I don't
I
know.
would say
So
not
far we've seen them in
necessarily,
pairs.
although it's been common so far.
And do grad students count the populations from the transect videos, or is it done by algorithm?
So for these transects that we're doing, we do have some contractors to NOAA. So Tom Leidig with the Southwest Fisheries Science Center has a team of folks that he oversees to do the annotation of these videos and pretty much the counting and the measuring. So far, we've not come as far in terms of counting fish and corals and sponges with algorithms, but I think we're on our way to that. I think that's where we want to be. Because Abby, who's been in the science chat and helping us out with our sponge information, has been working with a machine learning algorithm to count sea urchins so far.
So you guys see the tentacles on those clams there, and that's sort of the giveaway, key characteristic for keying them out.
Since we're heading that way, it's that close to a wrap on the 6-8.
Okay, let me get stretched out and then you can see what's going on better.
I think the tether should be fine because we're at half a wrap and it'll put us back at negative half a wrap.
As long as we don't put that loop in the...
Yeah.
That is.
You want to pause here or we can do this underway?
Do it on the fly.
Great. Ship moves underway.
There's a question here about when you're referencing voice communications, you're often referring to SPL, and what does that stand for? It might be a good opportunity to talk about sort of who's on the ship and who's not on the ship, and how all this communication is working between everyone.
Yeah, good point, Laura, and great question, listener. So SPL, I believe, stands for Science Party Line. So in terms of being on the ship, we have a couple of different lines that we can tap into. So if we just wanted to talk to the pilots or nav, we could switch over to that line in headsets, via our headsets. And we're currently, Julie and I are kind of virtually there through this software called V-Link. And we have access to two different lines, the Science Party line, which SPL stands for, as well as the Nautilus. So if we needed to talk just directly with the pilots to discuss our next ship move or what we're going to do about this certain C4 feature or a wall, for example, we could talk just to them. But the Science Party line goes out over Nautilus Live. And so we often refer to it to make sure that we're on the right line so everyone can hear us. We're answering questions and
Zoom in now.
And so Julie and I are participating from our homes and offices while the ROV
You're good,
pilots
Steve?
and the navigator.
Yeah, I guess that's.
sand
I thought it was
or
going to be more
sediment.
exposed, but it's still under the rock. That's fine.
Okay.
There was a comment about
And
the squat
video.
lobsters earlier in the dive.
Oh, the squat lobster there? Oh, okay.
It was exposed and then it jumped back into the rock. I don't know how much of the carapace you can see.
Retreat.
Yeah.
Yeah, it's very bright.
Okay.
And again, if we see any of those squirrel tail sea pens that seem to be attached to a rock, they're still interested in that. But so far, everyone we've seen has been anchored in the sediment.
Mm-hmm.
Squirrel tail, that's a really great name. It does look like a squirrel tail.
I know. I love it.
Oh, it's a crab. Never mind.
Is that a fish coming up there?
Zoom in on that.
It looks like a flatfish. So far, we've been seeing Dover soles. We'll zoom in and see what this one is.
Yeah, a little fin up.
It sure does. So it's a really good shot showing the two eyes on one side of its body. This would be on its right side. So when flatfish are larvae, they have eyes on both sides, and then their skull grows abnormally, and it pushes their eyes over to one side, and then they settle down onto the bottom. And then their pigmentation is usually just on one side. If you were to flip this fish over, it would be white or a creamy color on the bottom.
All right. Also.
But they're well suited to life on the bottom. And usually they do this. They just sort of lie in wait, and they can wait for something to come to them. This is what they do. This is the day in the life of a flatfish right here. Yeah. I would say this is a Dover sole again. It's hard to see what the lateral line's doing. That's another way to tell the different species of flatfish and any fish is you can look at the design or how the lateral line moves across the body from the gills back to the tail fin. But you can see there we go. Yeah, see the lateral line there? You can see it's a series of pores that runs along the body there, and it looks pretty straight. I'd say it's a Dover sole. Yeah, and you see the mouth is sort of oriented down, so they can, when they open it, they can actually pull things out of the sediment to feed.
Yeah, Erica Burton in the science chat is confirming that this is a Dover.
Thank you, Erica.
All right, I have one final question and then we'll wrap up the questions for the day. And that is, have you ever seen something that has really shocked or surprised you when exploring the deep sea, and what was that?
So far as a person new to this, studying this kind of environment, lots of things surprised me. Shocked, I haven't seen
right? So, that is? anything yet that's shocked me
anything
other than
I am.
Zoom in now.
surprised that we do see as many evidence
Looks like line.
accounts of evidence of human activity.
Sure.
For example,
Yeah, there's monofilament there.
yeah, we're seeing monofilament.
Oh, there is
to the left.
monofilament
It's
there.
kind of
Those look like the remnants of a
tangled in
kelp
this
holdfast.
seaweed. Yep.
So
That's kind of
the line
shocking.
they
It's
got-
such a remote
It got
system.
hung up in that.
In the holdfast, yeah.
Yeah. So there's another example of marine debris in the form of fishing tackle, monofilament fishing line, which we do see commonly on reefs where fishing activity occurs. And as scuba divers, we always carry a knife with us. And this is one of the reasons to carry a knife, is because you do not want to get tangled in monofilament fishing line. Just like an ROV doesn't want to get entangled in, either do people. When I'm diving, I usually remove this from the water.
So Herc has two knives aboard. And using the knife is how he got out of the first batch of fishing gear.
Yeah. And so this kelp holdfast is from much shallower. It's not normally found down here. It's sunk down to this deeper part of the reef.
It's that bright white thing behind it.
Well, thanks so much for taking the time to answer these questions, and we'll be back tomorrow and Sunday from 9
Thank you, Laura.
Again.
It was fun.
Yeah, thank you so much.
Great questions.
Yeah.
Oh, here's a Budweiser can.
Story checks out.
Okay.
It's an old
The story checks out. Monofilament fishing line and a Budweiser can.
Yeah. Looks a few years old.
Sounds like fisherman.
Mid-90s, maybe.
Yeah. Alrighty, well, science, we are at that supposedly flatter portion on high pack.
A little bit of flat.
Yeah. Go for it. Would you like to look around here at all, or start a transect over to waypoint X?
If the pilots are comfortable doing a transect towards waypoint X, that would be great. If not, we can just start going upslope towards that yellow patch of slope.
Probably deal with point 3 here.
Okay, great.
What's the bearing?
Let me get here for a second, and we're looking for a 200-meter transect.
Yes.
Great, let's do this one here. 312, please. Bridge nav. (200 meters) bearing 312.
Yeah, so pretty much after we get to waypoint X, Y kind of goes up in depth a little bit, but I'm wondering kind of like longer term, if we stay along this contour, depth contour towards Z, it's right around just slightly less than 500 meters in depth.
Ship move has started.
Line up our camera action.
Yeah, let me know when you're ready to start.
Maybe
like
need junk zoom in that? I don't know.
This looks like transect 5.
Right.
Thank you.
So for those of you that are just joining us, we conduct transects every hour. And they're usually 200 meters in length, and we follow a specific heading and take note of everything we see. We're not going to be zooming in on any particular species we come across during the transect. We're just going to be moving along and recording the sediment type, whether it's soft sediment or we're seeing some rocky relief habitat, high relief or low relief. Any species we see along the way could be a variety of sea stars or rockfish, sponges, corals. We're just going to document things as we go. And it's a systematic way of characterizing a habitat without putting a lot of biases into it. So even though we might see something off on the side that looks really neat and we want to go over there instead, we're going to stay on our course and only record things that we see in front of us along the transect. And this style of sampling is typical of what we do in science. We do it in every habitat, not only the deep sea using an ROV, but we can also do it with scuba dive surveys or out in the rocky intertidal. It's a way to sort of take a sample of an area and get a sense of the biodiversity that's there.
It's a great description of transects and why they do them, Julie.
Thank you.
Because it is tempting to just steer towards all of the really neat stuff we see, which in a given area, if we just recorded all that and said we transited this area, it'd be a really high density of those things that we really wanted to see. But in reality, if we were to look, kind of zoom out a little bit, the density would include probably a lot of empty, muddy areas like this that wouldn't have been counted in our video frame. So this is definitely much less biased, but sometimes
yes.
a little less interesting, but that's part of it. Zeros are data too.
That's right.
And in about 30 meters, we will be hitting a little bit of a downslope on our left, so it might get interesting with rocks.
Oh, okay.
That's a beautiful fish. Can't tell what it is from here.
Close up.
There you go.
Cue the close up. I'm not sure if the fish could tell what we were either.
God, he must be blinded by our lights.
So we're seeing more of those squirrel tail sea pens and some stars. Those little white dots, are those shell fragments or could they be just, okay.
I'm gonna guess they're probably shell fragments, probably mollusks, but it's hard to say for sure. I'm sure Daniel at the Santa Barbara Natural History Museum might be able to tell us, or would even potentially want a scoop of this. So we'll keep him in mind as we're finishing up our transect to see if we're still in some of this similar kind of area. Take a sample for him. Is that same fish, the shadow that we're seeing?
I think it's stalking Herc right now.
Lurking, just lurking in the shadows.
I wonder if there's anything worth
eating. I wonder if there's anything worth eating that fish is after.
Well, yeah, I wonder what's attracted to the lights, that it's sort of thinking maybe there could be a snack in there.
Yeah, I'm wondering why else he would get something so close to something so potentially obnoxious. If it's not very light down here, suddenly these blinding ROV lights show up. I gotta turn on see the Argus cam. What's going on? See if we can see him.
We keep seeing it. Oh, there's a couple. I keep seeing shadows. It's kind of kind of spooky. Goes with the fall theme. What's lurking in the shadow? Oh, very cool.
So cute. Oh, small guy. Yeah, I can't really see him in the Argus cam, but he might have left. He or she or it might have left. So it's interesting. We've been seeing on this dive just intermittent muddy slopes where we have a lot of these fragile pink sea urchins and these Anthoptilum sea pens and Amalula sea pens. And then we'll get into a rocky habitat again where we're seeing lots of sponges, some really small corals, but not a ton. And it's just kind of going back and forth between these two habitats.
Do you think that could be a type of a sablefish we're seeing?
It kind of looks like one, but I just am not good at my fish
I know we need
IDs, so
Tom
I didn't
to
wanna...
log back on. But I'm just
Why
reading
do
about
people
them a
need
little
to
bit.
take breaks?
We're in the right habitat for them. Definitely.
Yeah.
And they have quite a wide range. Japan, Bering Sea to Central Baja.
And they have a good range in depth too, right?
They do.
Like
Yeah,
feel like
they can
we've
go from
seen...
1,000 to 3,000 feet down to 6,000 feet or deeper.
What was the top of that range?
It said 1,000 to 3,000 feet, 305 to
Oh,
914 meters.
Okay. Well, we're at roughly 500 meters, so maybe he was looking, he was confused.
Yeah, I'm just going through my fish ID book, and it just, the way the head looked on it and the caudal fin, it was hard because we were looking straight down on it. I'd like to get a side view. If it comes back in, it needs to model a little bit differently for us.
Yeah.
We can try and
we
get
got
some
a
more
close-up of
distinguishing
its
characteristics of that fish.
head and its eye. Oh, is that a black coral? Maybe? On transect.
I know. Stay focused. Watch the green dots.
Yep. Oh, we had an eel, snakehead eel pout that we just passed just barely on the right.
The snakehead
Easy
eel
to
pout?
miss. Yeah, we just saw just a glimpse of its head and its pectoral fin. It was pretty much totally off screen.
Okay, so that's always a question. Do you count it or not count it? It is in our view.
Probably not. They might note this species, but I think with Tom's group, what they're doing is, especially with measuring them, I think they have to come very close to the lasers to be able to measure the size. So they might take a note of what they see, but I think that's different in terms of what gets counted and measured.
Okay. Is that one of those jellies that's parked on the bottom there, or was that something different? Or was that was probably a squirrel tail sea pin, huh?
Yeah, like an odd perspective, yeah, potentially.
Well, we're over right now the portion of the map that suggested there might be a slope with some more interesting features on our port side, but looks more like we have a light slope on our starboard side, so.
Yeah, I'm wondering, maybe we're just too far above it.
Possibly.
Like we're not really looking port-ish.
This is the thing about transects, right?
Yep.
We'll never know.
We never know. Because on the Bathy, it did look. Let's go back there.
Can't miss what you don't see.
Yeah, it looked like on the Bathy, it looked like there was some.
Interesting.
Yeah.
You tell two different stories.
Exactly. Because the flatter area we went over also was not quite as flat as we thought. So.
No, there was a brief flat part, like right at the lip of that slope, but then yeah, that was it.
I'm not sure those contours look 100% natural either.
Yeah,
There's some artifact in there.
Yeah.
might be right about that one.
Another bottle.
Oh, marine debris.
Corona.
That looks like a, I was gonna say,
Oh,
it looks like a Corona.
Yep.
There's a Dover sole. Is that kelp there?
Yeah. Oh well.
Or is it a?
We're approaching more sediment, like in between these areas that look like they have slope. Although this is very slopey, the high areas of high slope tend to be a bit rockier, but we see a lot of this kind of habitat coming up in on the HYPACK screen, I think too. And if anyone else is interested in looking at what we're seeing as we're deciding which directions to go, HYPACK is on channel 3, and it's also in the lower left-hand corner of the quad view on channel 4.
Yeah, when we're done with this transect, we will be just downslope from this steeper slope region on the way to X. Is that are these the contours that you'd like to follow that are currently kind of in the yellow area on the slope map?
Yeah, like I kind of want to stay on the deeper side unless that's really uninteresting. We can kind of follow it more upslope as we go across, or however best it is to transit
Sure.
that area.
Are you thinking, I'm going to drop a little target in like this contour line then, staying deep?
Yeah.
Great. Yeah, this area on the left also.
Wondering what that is.
Kind of interesting. Might also be an artifact.
Could be.
Oh, right there. Hmm.
Your maps are so pretty. I can't see any of this in my map. I'm somewhat new to ArcMap. I've only been using it for a handful of years, and that's really kind of to do some offhanded tasks. I don't use it to make really pretty high resolution maps on the regular, and this has been such a learning experience trying to plan these dives and provide maps that are informational to me to plan the dive, but then also to you all on the ship to give us the best information available. But it's been interesting seeing it on my ArcMap and then what you have in HYPACK, and it just kind of blows my mind. I'll get there one day. But I was really impressed. I don't know if anyone was watching this morning, given we're the 8 to midnight shift. I doubt it. I hope you were all sleeping. But the habitat that we dropped down on in waypoint U, which is kind of in this really nice hard habitat, which is actually fairly flat. There's definitely some ridges there, but in between here and Piggy Bank Reef was this magnificent, magical sponge reef. They're just huge sponges recovering, and then so many different species. It was absolutely amazing. I would love to go back there again. And we actually have, maybe we can update the next dive since this one's quite a long one. We'll have some time to think about it. There's also a bit of hard bottom habitat that looks kind of similar, but it's a little bit deeper, that goes to the northwest. And it kind of actually makes this ring, footprint reef, and then it comes down to meet the bridge that comes to Piggy Bank and then heads northeast to come back up to footprint. It makes kind of like a big square. And the backscatter is showing this all as hard bottom returns. It's fascinating.
So it could be promising.
It could be.
It could be.
So much promise.
Well, we are in this is a pink urchin barren.
Sure is.
Yeah.
So it's interesting. I mean, you think about these sorts of sediment dwelling urchins, and very different lifestyle than our shallow water urchins that are found within the kelp forest, shallow water kelp forests and rocky reef communities. They mainly feed on drift kelp and graze on the rock surfaces for algae. Down here, I imagine these are feeding on deposits on the bottom.
Yeah, you'll also find them clumped up on a big piece of kelp, a drift kelp that comes down here. So I think
so they're opportunistic.
When there is no kelp, yeah, when there is no kelp, they're probably feeding on very tiny pieces of organic matter from above. And then when they get like a payload, oh,
we're looking through the Argus
feel free to stop.
It's
camera.
Transect or no transect?
We've got 50 meters left on this transect as well.
Okay.
Well, I know there's another sort of a sediment-dwelling urchin too that's shallower, the Lidochinus, which is more of a white-gray colored urchin. And there's also the heart-shaped urchins too, that definitely are adapted to life in the sediments and the shallow water.
Yeah, it's interesting the number of habitats that you find urchins. But I guess the same goes for sea stars and brittle stars as well, because they have a bunch, tropical, temperate, all the way to, you know, Arctic, Antarctic. They're pretty much everywhere. Echinoderms are a huge group of animals that are extremely diverse.
They sure are. Yeah, and the urchins up in the shallower depths, the red urchin in particular, is what is targeted for the commercial urchin fishery, and divers go out and harvest those off the rocky reefs and around kelp forest environments. What they're targeting is the uni, the gonad material inside the urchin. It's a delicacy. And if conditions are right and they're getting well fed off of the drift kelp, then the gonad material is of high quality, and it can it's something that is sought after. A lot of it is imported from our out of the United States, but we also have some local fish markets that and it's sushi restaurants that feature uni on the menu.
Have you had uni, Julie? Do you like it?
I have not had it. No. Have you?
So my husband free dives and catches lobster, and every now and then he'll get some urchins too. And I don't know, he cracked one open, and I just couldn't do it. A cat shark just went off to the left.
Oh, nice.
Little cat shark. Yeah, I mean, I've heard that just like between the texture and what people say it tastes like, it's just kind of like salty and slightly.
That's what I've heard too.
Fishy.
Yes.
It's just not something I'm super interested in. Although it would be good to get like a healthy fishery for these to help in on the years that we don't have super healthy kelp forests that harvest. That looks like a dead sponge, maybe, with another sponge going on top of it. Either that or a big piece of trash. It's hard to say.
Definitely an outlier.
Yeah.
So how much more do we have to go on our transect?
We've got another 24 meters.
Okay.
Yeah, almost there. Little peak of a rock there on the right.
Is that a sign of things to come?
I think so. I think once we get to this higher sloped area, we'll see some more rocky outcrops. We're definitely in an area that's kind of mixed returns on the backscatter. once we get closer to the following waypoint, Y should be on a area with harder returns in the backscatter maps. But it's definitely, it has been an interesting mix so far.
And it seemed like there was a little sculpin that was sort of in our view. Ooh, there's a squat lobster. And a rock.
Sea star. Getting closer. And so kind of like what you said before, Julie, it's always great to to ground truth these measurements of the sea for the bottom of the ocean, because they're acoustic measurements. And so in terms of identifying hard bottom habitat and interpreting the strength of the returns of some of these acoustic blasts that we're sending through the water column, this could indeed all be hard bottom habitat underneath this slope, but there's just such an accumulation of soft habitat that corals and sponge can't grow here other than sea pins. Snailfish, another cute.
There it is, the black-tailed snailfish.
Oh, I'm so tickled with the zoom that we got on that snailfish earlier. That was just fantastic.
That was beautiful.
That was gorgeous.
Yes. And it lasted a long time too, so that was nice. Sometimes they dart off, but that one really was just hanging.
That one seemed like it was
happy to be here.
Trying to figure out what was going on there.
Yeah, I've definitely never seen it so iridescent like that.
Yeah, it was really
when
beautiful.
they're up
Yeah, I
close.
agree.
Yeah. And their pectoral fins are so interesting. They're very round, iridescent, and pretty much like under their chins.
Mm-hmm.
Would be interested in learning more about them as to why they've evolved the way they have.
Kind of remind me of a goby a little bit, right? Because they form sort of the suction, their pelvic fins turn into sort of the suction cups to attach to rocks and things.
The ship is stopped,
That's
you said?
point.
And what's next?
So, science, the ship is stopped, and Herc's got just a few meters left to complete this transect. Do we want to look around here, or just continue up to this target that we've dropped to follow that contour over to X?
Can we sample some of these urchins for Taylor Ann and take 2 sediment cores, and then I think we can proceed up to the target that you had dropped, Sam?
Sounds great. So transect is ending right now. Perfect timing.
All right, Al, let's get a zoom.
Just in time. We're taking some urchins and 2 cores.
All right, let's come wide.
Also, shoreside Paul is on Herc now, as you might have guessed by the
How's it going?
voice of Zoom.
By the what?
The voice of Zoom.
Yeah.
I am Zoom.
Well, technically Al is Zoom, but the voice.
Oh, I see.
How many urchins?
If we're gonna go push cores, do I go full throttle down?
Yeah.
A lot of throttle down.
Yeah.
And how many urchins do
I
you
think
want?
she wanted, Steve, you can correct me, but 4 or 5, I think. Does that sound right?
Yeah, I think up to that amount.
Yeah. I think she wants a minimum of 2. But I think if we could just get her the handful that's within reach.
How do we
Probably 4.
sample urchins?
One method that was useful was slurp and drop into a bucket. We can do that in the forward bio box, or you can just pick them up if you can get them gently.
There's not a risk of them being too small and going up the slurp and getting stuck?
These, I think, will be too big for the slurp, so they should just fit and nest right into the nozzle pretty well.
Okay. Well, let's go with the slurp and drop. That sounds good.
Yeah, so that we can go into the forward A box.
Confirming that this will be sample 113.
114. 113 was the orange heterocone.
Got it, thank you.
You on
That
grip
is.
force 9? Might not be. Might have been turned down.
Grip force 9. That was really slow.
Yeah, so probably it says grip force right there.
Yeah, that's 9.
Oh. Huh. Yeah. Well, that's a bummer.
I think maybe these here might be the best candidates to start with. Can we zoom in to see if how big they are compared to the nozzle size? 'Cause if they're small enough, I just don't think they'll get through the slurp though. I think they're definitely going to be.
You can just go for the bigger ones, I guess.
Yeah.
Yeah.
Maybe skip
Play it
this
safe.
one on the right and go straight.
Yeah, go for the bigger ones.
And so we're collecting these for the microplastic study?
Yes.
Yes.
Oh, what's happening on the... Can you zoom out? I don't... Either it's slipping...
Yeah, it's just slipping in the
Okay.
grip. Slip grip.
All right, can we zoom back in? Okay. Are we on a flush jar? Just to confirm.
Hang on.
Yeah. All right.
You want juice?
Yeah, let's give it juice.
That's 50%.
Oh.
It's on there.
Oh yeah, it's on there. All right, let's come wide out.
You ready for drawer out?
Oh.
Well, nothing you can do about it.
I think ready for drawer. Okay.
Ready?
Yep.
All right. No suction. Oh, there. There it goes. It's coming.
That's one.
Nicely done.
Yeah. Oh no.
You want to back out?
Yeah, let's back it up. I can't see what I'm gripping.
Yeah.
Maybe let's bring it in a little bit.
Coming in.
What's the diameter on that nozzle? Do you have a value on that?
Probably 1.5 inches.
Yeah.
So Erica Burton in the science chat, she works for the Monterey Bay National Marine Sanctuary and does a lot of their deep sea work up there on the Central Coast. She's letting us know that there are some fun facts next time we see a snailfish that we can share. The small pelvic disc that we saw, which are modified pelvic fins,
Which one do we want to go for next? Maybe that front one?
Yeah, I'm going to pull the tool.
Yeah, that front one looks pretty big.
We have an awful lot of background noise on P-Link right now. Be careful with our mics, please.
Okay.
Thanks for the heads up, Al.
You see it?
Now we hear a lot of typing.
Yeah, we got it. All right, let's come wide out. Okay.
But those snailfish, they have pelvic fins that are kind of adapted. You saw maybe if you were watching earlier that they're right underneath their chin, essentially.
Okay.
And they attach to tanner crabs
Is it suction
and
off?
research equipment,
Yep.
which is kind of fun. Researchers also found that they laid their eggs in the gill cavities of crabs, which is really cool and kind of disturbing for the crabs.
All right, what's our next target here? Maybe.
Just to the upper left of that last one, maybe.
Okay. Can we zoom in a bit, Al? Okay, can't really see it, but okay, let's come wide, I think. We'll try out. Let me straighten out. Okay.
Are we collecting a total of 3?
This should be
Of
number
the
3.
pink urchins?
We were going to aim for up to 5, but I think at least if we
Oh,
get
up to
2.
5.
Suction's off.
Yep, suction's off. I think you kind of bumped it, might have pushed it.
Oh, might have pushed it off.
Well, you might have pushed it in. It's past the lip.
Is there a reverse?
Shoot.
So for those of you that are tuning in, we are, Herc is equipped with a slurp hose, and they can create a suction so we can collect specimens. In this case, we're collecting pink urchins. And there's interest in studying these for microplastics, which has become an emerging issue in all of our ocean basins. And so we're looking both at microplastics in the sediment and also in urchins.
Can you rotate it so we can look at the end without ripping the hose off?
Not with this grip, maybe?
Yeah, no, I think you can get it, can't you? Other way, other way.
Some of the challenges we have in sampling in these deep depths is that we're doing it all remotely. The
keep going.
Hercules is down 519 meters from the ship, and we're relying on the manipulator arm. It's not like you can reach down and just sort of fix it on your own.
I think it went past the lip, so it's in the hose. That's not so cool.
Nope.
So now we have an urchin stuck in the hose.
Yeah. We could give it the gusto and see if it goes through.
What's the gusto? Is that like just a...
Turn the...
Do you push a different button?
No. Turn the pump all the way. I wasn't turning the pump all the way, we were just trying to get a hold of it, so.
Well, it's also close the bio box, see if, some chance it fell.
I think I saw it like...
Go in a little bit.
Yeah. Is it just crawling in? Is it it's just going you don't want to go?
I did see one float. I thought it was there was one floating while we were stuck.
I think you just kind of bumped it and it went past the end of the nozzle. At the very end, it's like a little bit narrower than the rest of it. So I think if you get it past the lip, then, it can get into the hose. If it gets past the metal bit up front, then the hose is bigger in diameter. So
yeah.
So it's possible to make it all the way through, but it's also, likely to get hung up somewhere.
Yeah.
Give it 100%, see what comes through.
Okay. Give it the beans. Sorry, actually, where does gusto and beans fall?
I think they're equivalent.
Okay, gusto's not more. Are they?
Oh, there it is in flush.
So yeah, see. Went through.
All right, so you want to try and slurp one more?
But not with full beans or all the
Ah.
gusto.